Have you ever just looked around at everything and wondered, how? How did we get here? I mean, it's the ultimate origin story, right? Not just for us, but for every single living thing on this planet. Well, today, we're going to try and answer that on a journey that's literally billions of years in the making.
So let's start with a central mystery. Just try to picture the early Earth, a chaotic, fiery, hostile place. All volcanoes and storms, no life whatsoever, just chemicals. So how in the world do you get from that to, well, this? Okay, let's dive into our cosmic detective story.
You know, before we could really piece together the scientific clues, people had some other ideas. So for this first part, let's look at the earliest attempts to explain where life came from. It really sets the stage for the scientific revolution that was to come.
Way back when, the explanations basically fell into two very different camps. On one hand, you had the theory of special creation, the idea that all life was brought into being by a supernatural power. But then, on the other hand, you had this really wild idea called the cosmozoic theory, or panspermia. This theory suggests that life didn't even start on Earth at all, but hitched a ride here on asteroids or comets.
But what if the answer wasn't from up above or from deep space, but was right here all along, cooked up in the primordial chemistry of the early Earth? This brings us to a really groundbreaking idea, the Oparin-Haldane hypothesis. And this, folks, is where our scientific investigation really kicks off. So the core concept is something called chemical evolution. It basically says that under the right conditions, life can emerge step by step from non-living stuff. Think of it less like a single magic moment and more like a chemical recipe for creating life from scratch.
All right, so what's in this recipe for life? Let's break it down. Step 1, you start with the primitive atmosphere, hot and full of basic elements. Then you let those elements react to form simple molecules like methane and ammonia. Step 3, you mix all that in the oceans, creating what's called a primordial soup, and you zap it with a ton of energy from lightning and UV rays. What you get are complex organic compounds. And the final step? These complex molecules started clumping together, forming these tiny, self-contained bubbles, the very first precursors to actual cells.
So a recipe is one thing, but could you actually prove it? I mean, could you really test it? Well, in 1953, two scientists, Stanley Miller and Harold Urey, decided they were going to try to cook up life in a lab. And believe me, this experiment was a game changer.
And here it is. This tangle of glass tubes and flasks might look complicated, but the idea was simple. It was designed to be a miniature early Earth in a bottle. They were literally trying to recreate the conditions of our planet's first moments to see what would happen. So, what did they put in their bottle? Well, they used all the ingredients from that Oparin-Haldane recipe: methane, ammonia, and hydrogen for the atmosphere, and water for the ocean. And for that jolt of energy, they used electric sparks, acting as tiny lightning storms.
They let this experiment run for days, just letting the mixture circulate, get zapped, and condense over and over. And then, something happened. A simple observation, but a profound one. As the notes from the experiment state, quote, "The liquid collected in the U-tube turned brown."
So what was this brown stuff? Well, when they analyzed it, they found it was a rich soup of organic compounds, including several different amino acids. Now this is huge. Why? Because amino acids are the literal building blocks of proteins, and proteins are the little machines that do just about everything inside a living cell. Urey and Miller had shown that the fundamental pieces of life could just appear from the basic chemistry of a lifeless world.
Okay, so we've got the building blocks. That's a massive step. But how do you get from a few simple cells floating around in a soup to the absolutely staggering diversity of life we see today? You know, from bacteria all the way to blue whales. Well, that question brings us to the next giant in our story. And here he is, Charles Darwin. Now, his theory didn't explain how life began, but it did give us this incredibly elegant explanation for how it changes. He provided the engine for all of life's diversity, the theory of evolution by natural selection.
Darwin's big idea really boils down to these 5 key points. First, organisms make way more offspring than can possibly survive. This creates a struggle for existence. Within any group, there are natural variations. Nature then selects the variations that are most helpful for survival. And over huge stretches of time, this whole process can lead to entirely new species.
If you want to understand overproduction, just get a load of this number, 2.8 million. That's roughly how many eggs a single salmon fish can lay in a season. Now, if every single one of those survived, we'd be drowning in salmon. But they don't, of course. And right there, that's the struggle for existence in action.
And this brings us to the engine itself. natural selection. It's not about being the biggest or the strongest. Not really. It's about having the right traits, the right variations to survive and have kids in your specific environment. It's often called survival of the fittest. And what that really means is survival of the best fit. Nature preserves what works, and what doesn't disappears.
So for a long time, Darwin's idea of slow, gradual change was the main story. But science never stops moving, and a new clue was about to surface. A clue that suggested maybe, just maybe, evolution could take giant leaps too.
Let's meet Hugo de Vries. In 1901, while he was studying these evening primrose plants, he noticed something really odd. Every now and then, a brand new type of plant would just pop up, totally different from its parents. And the change was permanent. It got passed down. He called these sudden dramatic changes mutations. And this chart here really nails the disagreement. Darwin saw evolution as a slow, steady, gradual process, tiny changes adding up over millions of years. De Vries, on the other hand, argued that evolution could happen in these big, sudden, random jumps. He thought a whole new species could potentially appear in a single generation from one massive mutation. So who is right? Was it a slow, gradual climb, or a series of big, sudden leaps? Well, as it so often turns out in science, the answer was kinda both. This realization led to what we now call the modern synthetic theory of evolution. And the modern synthesis is just a beautiful piece of science. It didn't throw out Darwin's idea at all. It made it stronger by weaving it together with the new science of genetics. It basically took Darwin's natural selection and de Vries's mutations and showed how they work together to give us the complete picture. So here's the bottom line. It works like this. Mutations, those random changes that de Vries saw, create the raw material, new traits, new possibilities. Then Darwin's natural selection comes in and acts on that variation. It's the force that decides which new traits are useful and get passed on. One creates the options, the other makes the choice. It's really the perfect partnership.
So, from a chemical soup to complex cells, from that very first spark of life to the incredible world we see around us, it's all one unbroken story. It's a story of chemistry, of chance, and of a whole lot of time. And it's a story that's written in the DNA of every living thing, including us. Which, of course, makes you wonder, what's the next chapter going to look like?